Yes, welcome back for once. I hope you are doing well wherever you are. Today we are still dealing with our topic, cell physiology, the 13th lesson in this series. Now, we had covered osmosis. We had looked at an experiment dealing with osmosis, 2 experiments. And before we look at other experiments, today we are going to look at osmosis in animal cells. So we are going to look at osmosis in animal cells. How does osmosis affect animal cells? Or how does osmosis occur in the animal cells? So our objective is actually to understand how osmosis occur in animal cells. So, to understand this, we are going to look at 2 different solutions. We look at isotonic solution, hypertonic solution, and hypotonic solution. So we know that animal cells are enclosed by a semi-permeable plasma membrane, and plasma membrane is also called cell membrane. So animal cells are enclosed or covered by a membrane which is semi-permeable. Semi-permeable in that it can allow selective movement of substances into the cell and out of the cell. And remember, osmosis deals with the movement of solvent molecules. And underline that, solvent molecules, like water molecules. So how does osmosis affect the cells, or how does it occur in the cells? Now, the cytoplasm of the animal cell contains dissolved solids. Which are these dissolved solids? The cytoplasm contains glucose, amino acids, mineral salts, and also nitrogenous waste. Meaning the cytoplasm of a cell also contains dissolved materials. So because of the presence of dissolved material, the cell normally exerts some osmotic pressure. Now, if, for example, we have an animal cell placed in an isotonic solution. Isotonic solution means a solution whose concentration is the same as the concentration of the cell cytoplasm. So if I have a solution, and then we place an animal cell inside that solution, let us say that is an animal cell. This animal cell has a cytoplasm. So this is the nucleus of this cell, and this is the cytoplasm. And inside the cytoplasm, we have glucose, we have amino acids, we have mineral salts, we have nitrogenous waste. We have all these things. Now let us say the solution that we have here, let's say the concentration of this water outside, or the solution outside, is maybe 50%. And the concentration of the cytoplasm, let's also take, is also 50%. So this solution here is 50%. Inside the cell is 50%. So what do we say? We say that the solution here, which we have, is isotonic to the cell cytoplasm. It is isotonic to the cell. Isotonic solution. So it is isotonic to the cell, meaning of the same concentration. And last time we said that what in osmosis, water molecules move from a solution of low concentration to a solution of high concentration, from a hypotonic solution to a hypertonic solution through semi-permeable membrane. But here is a case where we have same concentrations. We don't have high, we don't have low. So we have two solutions. The solution inside the cytoplasm and the solution outside the cell. They are separated by a semi-permeable membrane. Semi-permeable membrane, which is the plasma membrane. So what will happen is that if a plant cell is placed in an isotonic solution, then the amount of water getting out will be the same as the amount of water that will move into the cell by osmosis. Water will move out by osmosis, and some water will be getting into the cell by osmosis. And since the concentration of the two solutions are the same, the two regions are equal, then the amount of water getting out and the amount of water getting in will be the same. Therefore, because that movement will be equal, the cell will maintain its shape and size. So in that case, this animal cell will maintain its shape and size, because the amount of water leaving the cell will be the same as the amount of water that is moving into the cell. So one thing that we have learned from here is that an isotonic solution is a solution whose concentration is the same as that of the cell. So when the cell is placed in such a solution, the movement of water out and into the cell across the cell membrane will be the same. The cell will finally remain normal, and the size will also remain normal. That is how the cell, an animal cell, behaves in an isotonic solution. Now let's move to the second case, because we are going to consider three cases. One case isotonic, the second case now is hypotonic, and lastly we will move to hypertonic. So let us come back to our explanation. Now, so what about animal cell and hypotonic solution? What about animal cell and hypotonic solution? Now, when we talk of hypotonic, if this one is our beaker, then let's say we have our cell, the nucleus. And then we have the cytoplasm. Then we have the water, the solution. Let's say this solution is let's say this solution X is still 50%. Rather, let us use 30% solution. And then the cytoplasm, let's take the cytoplasm is 50%. So this one is lower, low concentration, lower, and this one is higher. The cell, the solution inside the cell, that the cytoplasm has a higher concentration than the solution in which the cell is placed. So if an animal cell, like red blood cell, is placed in distilled water, so let's use distilled water, and distilled water is 0%, pure water, 0%. So we are going to consider a case where we have pure water, and then we have placed an animal cell in that pure water. Animal cell placed in pure water. Then we are going to ask ourselves, what will happen? So that one brings us to the end of the first session. Let us meet in the second part so that we complete this.